All right, so hope everybody is having a decent Monday today. So I'm sad that we're not able to see each other on a synchronous session, but maybe some happy news for you is that besides this short video introducing cellular respiration, the only other thing I really want to make sure that you get done is just the enzyme catalysis lab for today. So hopefully you can feel a little less pressure to start your week here before we kind of get started. Just to let you know, the stuff that we're going to be doing here kind of this week and the start of next week on cellular respiration photosynthesis is usually kind of the more complex material that we deal with, especially in the first semester. So that's why I wanted to start out slow here today without too much that you could get confused with in our asynchronous session, and then we can really get to work here the rest of the week. So, today's just going to be kind of an overview and looking at how we harvest chemical energy in cellular respiration, because that's the overall goal to harvest chemical energy from nutrients and food that are took, taken in, into an organism and use that to produce ATP, like we talked about on Friday. So the whole point is to make ATP, right? That's the energy currency of the cell, that's chemical energy, and we need to make ATP, add phosphates onto AMP and ADP by harnessing that energy from other chemical reactions, exergonic, catabolic reactions, that give off energy, and put it into the ATP molecule. So the idea is that energy is stored in organic molecules, those macromolecules that we eat, carbohydrates, fats, and proteins. And heterotrophs, like us, that rely on other things, other organisms for food, eat these organic molecules by consuming other animals or other plants or other living organisms, and then digesting and breaking those molecules apart to get raw materials that we can use to synthesize different, more complex ones in our body, and fuel ATP for energy by slowly breaking these molecules down over a long period of time in a step-by-step manner. We can harness that energy and turn it very efficiently into ATP. Okay, and so all the reactions that we talk about in cellular respiration are controlled in individual steps by enzymes to help make the reaction as a whole, cellular respiration, very methodical and very controlled to minimize the amount of energy that's wasted. So when we talk about cellular respiration, we're going to use glucose as the model. So we're going to talk about how we break a molecule of glucose down to harness the ATP through the catabolic reactions of respiration. In truth, this happens with all types of macromolecules, carbohydrates, lipids, proteins, okay, nucleic acids, but there are some additional pieces of information, some added pathways for those, and we're just going to kind of focus on glucose unless you take like a more advanced biochemistry class specifically, because it gives you the gist of what's happening without convoluting it with a lot of details. So the overall idea in cellular respiration, you've probably seen this equation before, is that we take glucose or any other macromolecule in the presence of oxygen, and we go through a step of many enzyme catalyzed reactions to turn these reactants into ATP energy, water, and carbon dioxide that's just kind of a biochemical waste product, something that the organism doesn't need that's given off, and heat. And like I mentioned last time, this heat here that is given off in the cellular respiration is one of the main drivers for what keeps warm-blooded organisms warm-blooded. So the biochemical reactions in our body help maintain our body temperature. So the question is, how do we specifically harvest the energy from fuels by breaking it down? How do we harness that and make sure it doesn't all just go into heat or wasted energy? So when we digest or break down through hydrolysis larger molecules into smaller ones, we break bonds. And in breaking bonds, right, we release or remove electrons from one molecule to another. And those electrons from this point through the rest of this unit, I want you to think of as energy. That is the energy, at least chemically speaking, is these electrons. And so as we break down more biochemical or biomolecules, break down carbohydrates or proteins or things, we're removing more electrons from the bonds that are broken. And we uh, put them into electron carriers that carry energy with them in the form of stored electrons, right? And that those stored electrons are stored in another bond, okay, in that carrier molecule that we can eventually use to make ATP or release directly as heat in the organism. So this is kind of a simplified model. If we're breaking something down, like think of this as a glucose molecule, and we're breaking it into smaller parts, and we're removing electrons or energy from the bonds we are breaking, and we're storing those electrons in another molecule that is used specifically to carry those electrons to somewhere where that we can use them to make ATP. Now the terminology I want to introduce, or maybe you've heard before, is called oxidized and reduced. This is kind of like redox reactions from chemistry. Think of the glucose, the thing that is being broken down, that's losing the electron, as being oxidized. And the thing that's gaining those electrons is being reduced, the carrier molecule. And so one way I came up with that you can remember this is that Leo the lion says grr. Leo, if you lose electrons, it's oxidized. The lion says grr. If you gain electrons, you're reduced. So Leo the lion says grr. Hopefully that's something that you can use to kind of remember those terminologies. But more importantly is the idea of electrons we remove from bonds in a molecule represent energy. And whatever we're using to gain those electrons is carrying that energy to somewhere we can use it to make ATP. So we move electrons in a system by attaching them to hydrogen atoms. So electrons can't just move all on their own. Okay, so we're going to think that if we move hydrogen atoms, we're moving electrons, because each hydrogen atom has a proton and one electron that's kind of orbiting around it. So that is moving an electron. We're moving this proton and this electron together, so we're moving energy. So maybe a modified way of looking at the same picture here that's more realistic to biology and not chemistry is this. Okay, moving electrons, moving electrons. And so when we look at our cellular respiration equation, we have C6H12O6, that is glucose. And after cellular respiration, it's turned to CO2, 6 moles of CO2, okay? We've removed these 12 atoms of hydrogen. If we're thinking of hydrogen now as electrons, we've removed these electrons, this potential energy from glucose, and it has no more potential energy here. We add some of the hydrogen atoms, some of the electrons, some of the potential energy to oxygen to make water, right? But that doesn't account for everything, okay? The rest of it is used to make ATP. So the idea is that, like you might have heard about in chemistry, is that we couple that oxidation, that removing electrons or hydrogen atoms, with reduction, gaining those electrons and hydrogen atoms to make respiration work. So when we release energy as breaking down organic molecules, we break carbon-carbon bonds and we strip off electrons from carbon-hydrogen bonds by removing those hydrogen atoms, right? Think about, like we showed on the last slide, glucose going to carbon dioxide, right? The fuel, glucose, has been oxidized into carbon dioxide. Same thing like when you burn wood, it turns all into carbon dioxide and ash. The electrons we've removed, they've reduced another carrier molecule because, like we'll see, those carrier molecules are more electronegative and we'll accept those electrons. In biology, the most electronegative atoms are oxygen. So in this case, oxygen is the big reducing agent we say it gets reduced. So when we couple redox reactions and use released energy to synthesize ATP. So when we remove energy electrons from glucose and we add them to oxygen to make water, all right? So it's going uh, from these bonds, carbon-carbon and carbon-hydrogen bonds, and moving to oxygen, which is a lot more electronegative. We can use that released energy to make this ATP right here. So maybe a simplified way of thinking about it is this. Oxidation is when maybe you're adding oxygen to something and removing hydrogen. It constitutes losing electrons and a release of energy. So oxidation is an exergonic process. And reduction is removing oxygen or adding hydrogen to something when we're gaining electrons and storing energy. So it's an endergonic reaction. If right now you're seeming really confused, I might focus just on this page, because this is going to be the key information, right? The first step. If this seems like a lot, this is the first step, so maybe focus on this. So like I said, electrons or hydrogen atoms can't just move from one place to the other on their own. They need carrier molecules. Their sole job is to move electrons by shuttling hydrogen atoms around. And in cellular respiration and in photosynthesis, you're going to see these two electron carriers, NAD plus and FAD plus 2. When you see those, think of electron carriers. They're carrying stored energy and electrons from one place to the other. And so on the right, NAD plus and FAD plus 2, these represent those carrier molecules when they don't have any hydrogen atoms, when they can store more energy, store more electrons. And NADH and FADH2, look, they have hydrogen atoms. This means that they are storing the maximum number of electrons and storing the maximum number of hydrogen. So these have high potential energy, a lot of stored energy, and these have low potential energy. Okay, and here's two pictures, for example, of NAD plus, and how it stores energy in electrons by adding this hydrogen atom right here. No hydrogen atom there, hydrogen atom there. You don't have to be able to replicate this drawing. I just thought I could include it as a more pictorial way of representing what we're talking about. So, what we're going to look at in the coming days is kind of the stages of cellular respiration. All right, and we are going to talk about the aerobic parts, or the parts that require oxygen, which includes glycolysis. Or, sorry, I can't even read. The anaerobic parts that include glycolysis that can occur without oxygen. And we're going to talk about the aerobic parts, the ones that require oxygen, like pyruvate oxidation, the Krebs cycle, and the electron transport chain. So we're breaking down this whole equation for cellular respiration in 3 parts. And this is a diagram that can help you kind of figure out what we're going to do. We're going to talk about this part first and these parts second. Okay, You'll see this figure in your book, and also an assignment that we do tomorrow on Tuesday will have this as well. So we won't look at any of these today, but this is kind of where we're going to go from here. So that is where I wanted to stop. The rest gets into the first part about glycolysis right here. We're going to look at oxidation reduction in a conceptual framework tomorrow and talk a little bit about it more specifically when we come back together to talk about glycolysis on Tuesday. All right, I hope you have a good day.